Hi everyone, and welcome to Health Insight Hub, where we discuss important topics about health and science. So, today we're going to talk about the different methods that are used to study a cell, to study the components and constituents of a cell. And the word behind this science, the science of revealing components and constituents of a cell, is called cytochemistry. And under the umbrella of the cytochemistry, we have three methods that are cytochemical stain method, enzymatic digestion method, immunochemistry method. Now let's begin with the first method, the cytochemical stain. No, before that, let's break down the word cytochemistry so you can understand it better. So, cytochemical techniques make it possible to highlight in situ. And in situ means without extraction, in its natural environment, the chemical constituents molecules of the cell. So, let's move on to the cytochemical stains. For the cytochemical stains, uh, we use the cytochemical stain method. So it's the use of chemical reactions which will allow the identification of particular molecules in the cell. So let's break this method down. Let's define it more and give more examples. In simple words, let's imagine that we have colorful markers and we have a big cell in front of us. And every part of the cell should be colored in a specific color. And before coloring the cell, we have a manual. And in this manual, we have that every part of the cell or every substance of the cell should be colored in a specific color. As an example, we have the substance X should be colored in red. Substance Y should be colored in blue. Substance B should be colored in purple. This is the thing. So, for us to know that this substance is in the cell, we're going to color all the cell. With, like, as an example, we want to know that substance X is in the cell. And substance X is colored in red. So we are going to color all the cell in red, and after coloring it, we're going to have the results, and we are going to see in the results if we have the red color in the cell. So yes, there is the X substance. If there isn't a red color in the cell, there is not the X substance on the cell. So this is the thing. And it's the same thing, it's the same process for all the substances in the cell. As an example, I mentioned some examples here, like the detection of polysaccharide substances. Polysaccharide is a substance in a cell, in the nucleus. Detection of DNA by Flugen reaction. It's a reaction, it's the same, because when we color a cell, the substance makes some chemical reactions with the color, and it takes the color and it's colored. This is the thing. Let's move on to the enzymatic digestion. So, to increase the specificity of a cytochemical stain, the substance studied can be made to disappear by the action of a specific hydrolase. We then compare the serial section, example, detection of nucleic acids by the Brachet tests, and coloring methyl pyronine green VPM. Let's break it down. Let's give an example. So not imagine. Let's see it from a daily life perspective. If someone had a sugar, well, a high sugar meal, so his blood is gonna be on sugar high, and he should reduce the sugar on his blood, or he gonna fall in the hyperglycemia uh, phase. So to reduce the glucose, the sugar on the blood, we use an enzyme called hexokinase. And this hexokinase starts cleaving the sugar and breaks it down into small glucose molecules. And then we have the hormone, the really known hormone insulin, that helps us store glucose in the liver, in the muscles and things. So this is the thing. It's the same thing as it's an enzymatic digestion. We use enzymes to digest and to cleave big macro molecules into macro molecules into micromolecules. So this is enzymatic digestion. Immunochemistry. So let's give an example to simplify immunochemistry. Immunochemistry is like a detective game in the world of biology. Its goal is to find a special biological molecule in a cell or tissue using specific antibodies. We're still in the detective vibe. So, for immunohistochemistry, evidence comes from tissue slices, like pieces of a puzzle. But immunocytochemistry, evidence comes from individual cells, like examining single puzzle pieces. So the main idea here is we are looking for certain molecules called antigenes, like finding clues in our investigation. We use special tools called antibodies to help us. And for this immunochemistry thing, we have let's start with the principles. So, detection of proteins or other molecules, antigen, probes used, antibodies directed against the desired protein, and two methods. We have two methods: direct and indirect method. As we have two detective methods, direct and indirect methods. So for the direct method or the direct detective method, we attach a primary antibody directly to an enzyme or fluorescent substance. This method is not very powerful, like having weak clues in our case. So in simple terms, immunochemistry is a bit like a detective story where we search for special molecules in cells or tissues using specific antibodies. And there are different methods to do it, like direct and indirect methods. The direct method may not be very effective in finding the clues we need. So let's move on to the indirect methods. Indirect method or indirect immunochemistry method, we have two approaches. The first way, think of this as a two-step process. First, we have the primary antibody, which recognizes the molecule we're searching for. The primary antibody recognition of the molecule, and secondary antibody directed against the primary antibody, and is coupled with the following things: fluorescent molecule, an enzyme detected after incubation with a colorless substance which becomes colored after reaction, an electron opaque particle, or a radioactive molecule detected by autoradiography. So it's the same thing. As I said, so which recognizes the molecule we're searching for. Then we use a secondary antibody which is directed against the primary antibody, and this secondary antibody is like a detective's helper and can be linked to different things such as a fluorescent molecule like a colorful marker, an enzyme that becomes visible after a chemical reaction, tiny metal particles for special electron microscopes, a radioactive molecule that we can detect with a special imaging method. Then we have the second approach, the second way, amplification. So for the second way, we start with the primary antibody just like before. Then we introduce a secondary antibody which is directed against the primary antibody, but in this second way, the secondary antibody also carries something extra called biotin. So, to simplify it, in the indirect immunochemistry method, we use two different ways to find our molecule. The first way involves two antibodies, one recognizing the molecule and the other revealing it in different ways. The second way adds an extra molecule to the mix for amplification. It's like adding a detective's helper with a special tag. After talking about the different methods to study components or substances of a cell, now how can we study a cell without separating it from other cells? So now let's delve into the separation of cellular constituents. We have three methods: cell fractionation, electrophoresis, and chromatography. Chromatography. So let's start with the first method: fractionation of cellular constituents. Principle: it makes it possible to obtain after cell bursting and centrifugation pure fractions of organelles or macromolecules. Two steps. The first step is preparation of the cell homogenate and the separation of organelles. So, like imagine you have a cell and you want to study its different parts, like its organs, which is called organelles here, uh, different organs and thing. But first you need to break the cell open and make a mix of everything inside. So for the first step is preparation of the cell homogenate. So it's preparing the mix to make this mix, called a homogenate. We use different methods to break the cell's outer covering. These methods include using osmotic shock method, sonication method, mechanical grinding method. And as we said, the second step is sorting the parts. Like once we have the mix, we need to sort the cell's parts, like its organs or big molecules. This helps us study them separately. It's a bit like storing a pile of different toys so you can play with each one of its own. Yep. So the first thing is osmotic shock. So osmotic shock is a general way to break cells. Osmotic shock is like opening a delicate cell with as little damage as possible. How it works? So, cells are placed in a solution with less stuff, a hypoosmotic solution. Cells try to balance the amount of stuff inside and outside, so they take in water. Too much water causes the cell's outer layer to burst, like a balloon popping. This helps us get nearly perfect cell centers, nucleus, without the outer cell cover. To simplify it, osmotic shock is a general method to open cells. We put them in a watery solution, and as they soak up water, they pop open. This way, we can collect the cell centers without much damage. Such as an approach allows recovery of almost intact nuclei once separated from plasma membrane debris. Let's move on to sonication. So sonication is using sound waves to break cells. So it consists of the distraction of cells by ultrasound. So how it works? Sonication is all about using powerful sound waves to smash cell apart, like Hamza do. He smash people. Here we smash the cell. So with ultrasound for sure. We use a thin metal rod, which is called the sonicator, that goes into a cell mixture. This rod vibrates very vigorously. The vibrations create super strong sound waves that make tiny bubbles grow in a process called cavitation. These bubbles experience a lot of stress because of the powerful sound waves. In simpler terms, imagine using a magic vibrating rod to make cells burst. It's like turning up the volume on sound waves, which creates bubbles that get super stressed and help break the cell. So these bubbles are subjected to great stress by ultrasonic waves. Now let's move on to the mechanical crushers or mechanical grinders. Mechanical grinders or mechanical crushers, like the powder homogenizers, work by forcefully making cells move through a narrow space. How it works? There is a ball with a piston. We quickly move the piston up and down in the ball. This makes the cells go through a small gap between the piston and the ball's wall. The cell membranes open up. Because of this squeezing and the cell parts comes out. Depending on the cell size, we use different powders with varying clearance, which is the space between the piston and the ball's wall. In simpler terms, it's like using a powerful grinder to squash cells and get out the stuff we want. Different grinders are used for different sized cells. Yep. Oh, so I think our video comes to the end. So alright folks, that's a wrap on this journey into the microscopic universe of cells. Remember, like these tiny powerhouses are like the superheroes of our bodies, working hard 24/7. So stay curious, keep learning, and don't forget to hit that subscribe button for more mind-blowing science adventures. Until next time, stay lit, stay curious, and keep rocking the science scene. Peace out, my fellow bio explorers. Bye.